Yeah, so hi, thank you so much for for letting me speak and present.
Speak a little bit louder.
A little bit louder. So is it?
No mic here, it's just for recording.
Okay, so that's for, I see. Yes, so let me go to the introduction. And basically, I'd like to introduce myself and the center. I work at NORMENT. It's a Norwegian Center for Mental Disorders Research. It has 4 big domains. So we work in genetics of the psychiatric disorders. We work with brain imaging. That's another big domain. And there are 2 large clinical departments, which are like probably more than half of the employees at NORMENT working with antipsychotic medication and outcome prediction, right? So what we will talk about today is about genetics. So it's this part here. And the reason why I'm so interested in genetics is because we can apply lots of really interesting mathematical models to that data. And my background is, so it's like you said, it's Faculty of Medicine here. I'm actually like zero medical background for myself. I'm coming from the Moscow Institute of Physics and Technology. I worked in software for a couple of years, so just coding and doing some applied mathematics. My PhD with Vorontsov was in the theoretical backgrounds of machine learning, so we study overfitting. Like once we fit the model, it always performs worse in the independent set. And it's really interesting to bring this kind of knowledge into the medical domain and try to see what's actually wrong with a patient struggling from psychiatric disorders, because it's not nearly obvious. Whatever technology we apply, from DNA to brain imaging, the brain looks surprisingly normal. The DNA looks surprisingly normal. We really don't know what's wrong. So, but we know it's heritable. We know from twin studies that schizophrenia is highly heritable. There was a case when basically all 4 identical twin daughters were sick with schizophrenia, so suggesting that it must be some genetic component. And roughly 80% of schizophrenia risk is estimated to be due to genetics. So that's why we're interested. And so basically on this slide, I'm showing how we believe the complex human traits looks like. And complex here basically means polygenic. It means that a lot of genetic variants are involved in the genetics. You know, we are humans, so we have 23 chromosomes, 23 pairs of chromosomes. And on chromosomes, we have these genetic markers, right? It could be A, T, C, and G, and I just can't remember how you spell out the full names for these 4 potential nucleotides. And it's quite identical, right? So if you compare these 2 kind of synthetic figures, it's really identical DNA, except for this single location, right, on a chromosome. So here you see the, let me see the, where it's pointing to? Right, so it's blue and yellow on top, and it's green versus red in the bottom, right? So that's a point of DNA here when we call SNP. So SNP is an abbreviation for single nucleotide polymorphism. It's a point in DNA when different people can actually have different DNA materials, right? So this is a genetic variant that we are interested in. And like just imagine, okay, if we have 1 healthy person and 1 person who is schizophrenia affected, right? We could ask the question, is this because of this SNP, right? Is this specific SNP that is causing the disorder? But schizophrenia is not a Mendelian. By now, we definitely know that throughout the whole genome, each chromosome has some risk factors, right? So having a sample of 1 and 1 doesn't really work for us. We actually need a larger sample to like look for statistical significance. For like given that we observe some difference in, let's say, in the variant A, it's normally G in healthy people. So we say that controls, these are healthy people, right? We have to collect large samples, and we see that in healthy people, this variant is normally G. But if you look at cases, then it's another single nucleotide polymorphism that we encounter like 50% of the times. So probably this one has something to do with the schizophrenia. And if you count frequencies of different DNA markers in a given location, then we can, for example, use exact Fisher test in this case to see whether the combination of the variants you could have is statistically different between your controls population and cases population, right? So that's kind of by doing that across the whole genome, we can see, okay, these markers of some locations on the chromosomes are associated with a phenotype. And you can also think of this in terms of like naive Bayes classifier, right? If you treat these genetic variants as your predictor features, right, and your human trait as outcome, like schizophrenia or not schizophrenia, right, we can basically try to do a naive classifier which just independently looks at the features, right, and see whether this particular feature, what does it have to do with the disorder, and then kind of try to accumulate them together, right? It might not be even Bayesian, it can be any naive classifier that treats features one by one, right? So that's where we are, like here. And so far I've been talking about like differences in groups, right? We have healthy people here, we have people with a disorder in the bottom, but like for the rest of the presentation, we'll actually look with what people call quantitative traits, like human height. You know, tall people, tall parents normally have like, tall kids, right? It's definitely heritable, but like exactly how it is heritable, like it's pretty interesting. So in our case, the very good example of the output will be the human height. If you go to now it starts on mathematics at last. So yep.
What is the typical size of cases set and controls?
Yes, so if nowadays it's approaching millions. Really millions. So in case of, at least for example, the largest device on height is more than 1 million people, like because it's easy to measure. Schizophrenia sample is a very large international effort led by a Psychiatric Genomic Consortium, and the previous release, it was 43,000 cases. This year they will release something in the order of 100,000 cases. Because as you can imagine, we have 3 billion pairs. Not everything is, of course, involved, and like a lot of DNA material is similar across people, right? But still, the amount of points of DNA when there is a variation, even within the human population, in the order of tens of millions, and it's even larger in African population. But in European, it's if we count everything that is at least 0.5% of the people have a variation in a given DNA, we will end up with like 11 million markers, and like. Because it's so polygenic that nearly everything has something to do with the disorder, you can imagine what are the effects, right? So human height is probably between, I don't know, 150 and to like 150 centimeters to 190, right? So it's pretty small range for so many variants. I think the largest genetic variant known for height is like less than 1 centimeter of effect for that variant. Mm-hmm. And then the bulk of the variants, if you look at like 50% percentile, is probably like in terms of 0.5 millimeter or even less. It's very hard to detect like the association from those variants. So we need a very, very large sample sizes to get anything. The amount of noise in this data is kind of the general theme I would like to I can't stress enough how noisy is this data and how difficult it is to get any sensible readout out of it. So back to our height. So we have this n is the number of individuals in the data set in the order of tens of millions or hundreds of thousands of people. M is the number of genetic variants in the like about 10 times larger at this point, around 10 million genetic variants. The Y is our outcome, the phenotype vector or human height, right? And then so I have this both like. So I have a couple of formulas here. So on the left is just written down, and on the right is a matrix form. It's exactly the same, whatever you prefer to look at. So G is this genotype matrix. This is actually observed. Thanks to technology, it's not super expensive to get people genotyped, and we actually have access to these genetic features these days. So N individuals and M genetic variants. The beta is what we're really interested in. This is a vector of genetic effects, right? And then, of course, we have some kind of other non-genetic component, for example, environment or the non-linear part, right? So here for simplicity, but every research so far is on like complex human genetics is doing this linear model. So it's very hard to go for interaction between SNPs, between these genetic variants. The linear model is really like mainstream and good approximation to the. So it's not just approximation. It's also there are some biological reasons why we're only interested in linear modeling. So yeah, Y is known. Of course, we can measure height, and now we can like get the lab to genotype people or sequence them, and we know G, but we don't know much about the genetic effects, right? So that's what we're interested in. Yes. a little bit more louder. Okay, really sorry. Okay,
but
yeah.
You could use mic.
I don't like. I can try. Yes. Oh, so that's an easy fix. Yes. So once again, let me repeat because that's kind of a fundamental. It's called simple additive genetic model. We just model a linear relationship between genetic features and the phenotype, and a beta is unknown vector of genetic effects that we would like to find. Like we all know linear math, so like why don't we just kind of do that, right? Why don't we just invert the genetic matrix and try to? So one thing is we have 10 million by 1 million, right? It's pretty large, and even technically, I think that's difficult, right?
But it's also not square.
It's not square, so we can do pseudo inverse. Sorry, that was a typo. I can put it as typo. But also, it's like very low rank matrix, right? And the genetic features are highly correlated, and like the amount of noise is just too much, right? So what people tend to do is, as I mentioned, a naive classifier when we just look at one feature at a time. So if you introduce this genetic vector I mentioned before, right? So the genetic the genetic variance for different individuals for a given marker, right? So it goes from 1 to n. That's how many individuals we have, and then for a given genetic variant, right? That is the column vector that we have, and we can just see the correlation between that vector and the phenotype, right? Whether what you know from genetics about a given SNP correlates with your phenotype vector, and that's your beta hat, just marginal one SNP kind of association study. It's really like proportional to the correlation, the Pearson correlation between phenotype and genotype, and like it's a bit noisy. So we normally think like work in terms of the statistical strengths of association. So if it would be better to write the T statistic, right? Because that's a Wald test. It takes the estimate over the standard error of the estimate, and effectively it's like just the square n times the correlation. That's, of course, the larger sample size you have, the more statistically significant is your finding, and also the larger the correlation is, the better. Like the larger is the effect, the better it is. So, like, so far, really, really simple. But can we actually say that this beta hat, which is a correlation between phenotype and genotype, has what it has to do with the underlying vector of the genetic effects? Like, since I call it beta hat, you would probably think that it's actually like an unbiased estimator of the beta, right? But that's not the case because of correlations, right? Because our genetic features turn out to be really, really correlated, right? So maybe I can show this, go to the correlations straight away. So if we have these SNPs and then look at the Pearson correlation across SNPs, right? Then it will be huge blocks of up to like 2,000 or 3,000 genetic variants large. Because, you know, on a chromosome, things are inherited together. Like the large pieces of chromosomes, of course, there is a crossing over that we used to do in the genetic algorithms in like optimization methods, right? Inspired by like crossing over in genetics. Yeah. But yeah, in humans, it's a pretty large chunks that inherit from one parent. So the variants located close to each other tend to correlate. It's like this autocorrelation structure along the chromosomes. And it looks like blocks, right? So there are the hot spots where chromosomes tend to break. However, there is like a hierarchical structure to it. I'm sorry for the colors. I can't really explain what the colors here mean, but it's more like you do see that these blocks, they are not like, these blocks seems to be quite separated from this one, but you can also imagine that there is a larger block like of this size here. So it's a complex autocorrelation structure along the chromosome. And
the more block the color, the highest correlation?
So not in this case. I should redo the. So it's a bit messy, but yeah, it's yeah, best not to like just kind of do a black-white for this matrix. Sorry about that. That's just the figure I had. It's from a review paper. It's specific questions I asked, so not the best illustration here. As a result, if you go back right to what we like now we look at these z-scores, right, the correlation coefficients between genotype and the phenotype vector. It's possible to prove that they are again a linear combination of the underlying betas that we are interested in. It's kind of a theorem. So instead of working with the original problem, which was linear, we can just still work with exactly the same style of the problem. But it's now, before the y vector had the length was n, the number of individuals. Now we have an observed vector of the length m, which is a number of genetic variants in the data set, right? And now A is a rectangular matrix. It's m by m, right? We just looked at it on the next slide. It's a correlation matrix, how correlated are the genotype vectors. And so basically what we can kind of, I call it theorem because we can definitely, there is a proof that these correlations that we can measure, they are a linear combination of the underlying beta features. So the exact, the theorem is that the elements in the sparse matrix is some kind of a little bit adjusted genetic, a little bit adjusted correlation coefficient between the feature vectors. Could be a little.
So can we say that Z is less correlated than beta?
So.
Can you say that we're performing a kind of decorrelation, a kind of white?
Not necessarily. I think actually the correlation structure of Z matrix is very similar to the correlation structure of the features itself. It's more that instead of working with, okay, the main reason to go into that other space, like I'm trying to explain it, like why we are doing it and why we put two kind of very similar mathematical tasks, but at the end, like we will now switch to the next topic is a mixer model, and then just either of these form will be equally good to discuss what we are actually doing in terms of math. But the idea is that this was a domain of the sensitive data. The individual level data is very sensitive. No one wants people like the society to know their status schizophrenia or not. And then the genetics data is also sensitive. Like good luck anonymizing genetic data because it's like an anonymous fingerprint, right? It's the identifier, right? My DNA is me, right? So, but in this space, there is no more n, right? It's all about m, which is a number of features, right? And the correlation matrix, like nobody, people okay with like the society knowing the correlation between the SNPs in the genome overall, because it's not about a given person. So, and basically that's the main reason to go from this domain into this one. And we made the task probably a little bit more challenging mathematically because the design matrix was like 1 million times 10 million, and here 10 by 10. But it also became a lot sparser, right? Now this is actually just, I've shown you the block size, right? Just the blocks are roughly 2,000 SNPs at a time, right? So it's possible to store this correlation matrix highly efficiently because of it's a sparse banded matrix, right? And the exact shape how we transform from G to A, it's not so important, but basically that is like here the REG here was a correlation coefficient between the genetic features, right? So for two SNPs, if we have the vectors, then we can correlate them, and that's basically our A matrix with small adjustments. So that was basically like the introduction. What data do we have? We can observe Z and the design matrix, which is really huge, and then we are interested in betas, and we can't invert, right? I hope so. Like, can I probably do this trick? Could you please raise your hands if you understood what's known and unknown in this equation? Yeah. So that's good. I can probably reiterate it once more time because like as a whole second part would be about solving this. It should be quite simple because it's a system of linear equations, right? Let's now, I know it's very difficult to get in the domain. So like really don't worry if you didn't fully understand how that is connected to the genetics data. I just thought I could really start this talk at this point and say, okay, we have known Y vector, we have known design metrics, we don't have anything, like we would like to find the betas, right? And the epsilon, you don't even need to write it in because in the linear, like if it's OLS, like overly defined OLS system, right? Okay, it's just, that's a normally distributed residual, right?
So in some sense, it's all about solving a large-scale linear regression problem.
Yes, let's boil it down to that, right? And now we're going into the model that we've published recently, and the model is really simple. Nothing we can surprise you with. It's a mixture prior. It's a prior where we say that only a small fraction of pi1% of the variance have non-zero effect. So it's probably weird like this term is just a normal distribution with zero variance. I could call it delta function of Dirac, right? We just say that some variants, actually the bulk of them, have no effect. Their betas are zero, so it's a sparse. It's not only the A matrix that is sparse, but it's also the solution that is sparse as well.
Is it a prior distribution? Is it prior distribution?
So this is our prior distribution we would like to enforce on beta.
Just one clarifying question. Are you familiar with the spike and slab?
Yes,
because
yes,
that's exactly it.
Yes,
okay.
That is spike and slab. Yes. So yeah, and it's also zero mean, right? So that's quite important. That we don't know whether like our phenotypes are centered, right? I didn't mention that, but it was height, but of course we kind of center it, so it's zero mean, right? Now we obviously would like to write down the likelihood function in order to do the fit, right? So that we will spend some time. How do we compute the likelihood? Because so basically we are interested in computing the likelihood over like given z, right? Given our vector of parameters. And like the reason why it's difficult in this case, right, is because just imagine if you have a sum of two. Just tell me if that's obvious, right? But I want to explain that. If you have a mixture of two normal distributions, and now you do a linear combination, then now you actually the distribution at the end is a mixture of four normal distributions, right? Because depending on which branch it's coming from, each variable, right? So if you have 3, it will be 8, and it's 2 to the power of k, where k is the number of things in your linear combination up here. So given that we are talking about the like SNPs of like 10 millions or even the block size of 2,000 for a given LD block, it's just not tractable, right? To get an analytical expression for the likelihood function. So at this point, I would like to ask, like, do you know some theories? Is there a name to this domain when people have a mixture distribution and then they do linear combinations of these probability density functions? Must be some theories. If not, that could be an interesting theoretical piece when we study mathematically. We study an arbitrarily linear combinations of the mixture, two-component mixture of Gaussians.
There are some approximate methods for such kind of problems. There are some approximate methods for such kind of problems. For example, we can, what I could think of is we could try to reconstruct, say, approximate posterior distribution in some parametric family such as Gaussian and get something like assumed density filtering or expectation propagation. Something like this.
So let's compare that to what we've done, because we've spent 2 years doing that. Like doing something with this problem, because we really believe that this is a good prior. And I will, like, that prior is pretty simple, but I will show you, like, now we're working on the next paper where the prior is a lot more complex. And, like, of course, we just start with that. But,
again, just to clarify. So you're using prior distribution over beta, which is fully factorized, component-wise, right? And each component is a mixture of two Gaussians.
Yes.
Derive delta function plus some Gaussian.
Yes.
And if we factorize them, do we really obtain exponential number of Gaussians?
Yes.
I would think that we should obtain one Dirac function in multidimensional space plus one fully factorized Gaussian.
Not really. So because I can try to just put it. It's long enough. That's good. So where do we have markers to write on it? Yeah, this one. So let's just try to have it with no.
Let's
consider a normal
a two-dimensional case.
Yes. Yes.
Do you still have marker?
No, I don't have the marker. But we have desk. Because that's something would be nice to write down, because really I'm
afraid that there's no marker.
I'm trying to find it.
Thanks. So the idea is, let's say we have let's, for example, consider a sum of two random variables each having this shape, but let's assume that the pi is different. They are still independent, right? So they do factorize, but let's try a different pi 1 and pi 2 in the two things we put in the linear combination. right? And A will be equal to 1, right? Just this plain sum, right? So you're right. There will be one Dirac, right? With probability y1 minus pi 1 times 1 minus pi 2. There also will be the one with sigma beta 1 plus sigma beta 2. And that will be the probability pi 1 times pi 2.
Right.
But there will also be like 1 minus pi 1 times pi 2 and 1 minus pi 2 times pi 1.
Yeah, and there'll be product of delta function with respect to one dimension and Gaussian with respect to another dimension.
Yes.
Right?
Yes. So.
So kind of partly degenerate distribution.
Yes. Oh no, but it's still 1D, right? At the end, it's still 1D because each distribution is like a function of one. It's a one, right? We don't do it in 2D in this sense. So we sum them as a random variables.
But then I do not understand what would be the third and fourth multiplier, which is the product of delta function times Gaussian. Right?
It's a sum, right? It's so the way
if it's a sum, again, a sum of delta function stands at 0 plus Gaussian, right?
Yes.
Would it be just Gaussian?
That would be just Gaussian. The question is, depending on which branch it's coming from, branch of the probabilistic mixture, you have like the. So I think at the end it's very, very simple. If we have 1 minus pi 1 and pi 1, and 1 minus pi 2 and pi 2, and we are interested in 1 minus pi 1 times normal distribution 0, 0, plus pi 1 normal distribution 0 sigma beta. Let's call it sigma beta 1, sigma 1 even, just sigma 1 square. Right? That's one random variable. And we would like to add to this random variable something else, 1 minus pi 2 of the same one, plus pi 2, 0, sigma 2 square. Right? So the problem is the result so if we observe an observation from this branch, it still can come from either of these ones. So the way I can write, I mean, maybe we can write it differently, but the way I can write the result will be a mixture of all distributions with probabilities 1 minus pi 1, 1 minus pi 2 times normal, 0, 0, plus, right, so that would be this one, the product of, right, so then it will be 1 minus pi 1 times pi 2, meaning that it came from this branch and that branch, right, so the variance will be 0 plus sigma 2, so 0 sigma 2, right? Then another 1 minus pi 2 times pi 1. And this time it's sigma 1. And then the most interesting component, which is like pi 1, pi 2, and then sigma 1 square plus sigma 2 square. Right? And then if we add the third one, then suddenly we need to like consider all combinations, which branch of the probabilistic mixture it's coming from. So that's how it approaches this combinatorial.
And this will mean that the resulting distribution over Z will be a mixture of exponentially many
that was, yeah.
Okay, up to now it's clear. Although I still can't understand the last two lines on your slide.
So the last lines is if we can come up with some analytical way or the way to compute the distribution over z-score given our three parameters, then we can, like, because we observe the z-score, so z is observed. We have these numbers, right? So now we can do the max likelihood. We can. So
yes, but it seems that that's a bit unstandard problem formulation. Because up to now, well, maybe I understood something wrong, but up to now we established prior distribution over beta.
Yes.
And now we're trying to maximize our likelihood with respect to prior distribution over beta. Maybe this is just some.
So maybe I'm not using the So we will not introduce what like the Bayesian prior over the theta vector. That is so we do it in the like probably in the non-Bayesian sense, in the frequentist way. So the posterior.
What is the max evidence like?
Hmm?
It's max evidence.
It's I call it max likelihood because
We'll realize in a couple of slides whether that's maximum evidence or not.
Mhm.
Dmitry,
Yeah.
can you please explain why is it a problem with a lot of Gaussians? Because we have mixed Gaussian as a prior, and we have Gaussian likelihood. So shouldn't it be like the posterior should be like just the mixture of Gaussians?
Well,
Also
so-
two of them?
Not a-
Maybe I don't understand, I'm sorry. But maybe you can explain it in different
again,
points.
let us look at the first equation. So Z is a linear combination of betas, right? And now we know that each beta has a distribution which is a mixture of two Gaussians. One is degenerate delta function 0, and one is some zero mean non-degenerate Gaussian. And now we simply take linear combination.
Yeah.
If we take linear combination of random variables, each of which is a mixture of two Gaussians, we'll end up with a distribution which is a mixture of an exponentially large number of Gaussians, 2 to the power of m.
Mhm.
So that's it.
Yeah.
And we'll have some difficulties in computing P of z j
Yeah.
given theta,
Yes.
because this is a mixture of exponentially large
However,
numbers.
at this particular moment, there is an obvious solution. Let's just add some hidden variable to our prior distribution to say from which component we have our beta,
Yes.
and just run the variation of EM. Why this is not the answer?
So yes, I think that's one of the. So at this point we are not really interested at. We are only interested in this fitting these three parameters, right? So whatever way we can compute the P of Z given theta, right? So I think that's what we are doing actually. Like on the next slide, what I call sampling approach is exactly that. We randomly sample which of the variants. Right, so okay, let's just for simplicity. If pi1 is equal 1, then we have no problem, right? We no longer have the mixture. The result is just one single normal distribution. No problem at all, right? We just analytically, and that's the idea underlying the least score regression method. 3,000 citations on the Nature Genetics, really easy math. Lucky guys, right? What we know based on domain that basically it's a bad prior. The pi1 equal 1 is a bad prior. Like I've complicated the task for ourselves by saying now we are going to model with this delta function plus the normal distribution, but it does fit the data a lot better. So like unfortunately that one of the priors that we know is kind of working for our data. So let's work out the technicalities. And we thought that it's a nice idea to just do a sampling, right? Once we can guess, like select, let's say, one subset of the variants that are coming from the pi1 branch, right? Then we can do the nice trick with pi1, right? So then it will be the, so here, if we know the set of branches that contributed the variance to the result, then we can compute, kind of in this fashion, we can compute.
Can we be sure that this is unbiased estimate?
Well, what we know is that it does work. In terms of the pure statistical properties, I guess we never formally prove that it's an unbiased estimator. But in terms, like we can compute the expectation of this thing, right? We know that the expectation is correct. The expectancy of the z-square distribution will be okay. Whether it like all moments are preserved, that I don't know. But at least the first moment, the second moment is okay.
Okay.
And in our simulations, we have to do around 20,000. So if we 20,000 times sample from the set of potentially of the non-zero betas, and so the difference is AYG, right? That is different across variants.
Yeah, sure.
If we just sample a subset, and then we can use kind of this simplistic formula. And by the way, could you tell me how to write it properly? Because like this sum is a mixture. It's a mixture of capital T distributions, each with either equally weighted, is equal weight, the 1 over T, and then I actually go for like normal summation. So I hope that's it's a pretty large mixture distribution.
Okay, this is mixture distribution.
Yeah.
We know that the mean will be the same. It will be 0. Right?
Yes.
Just as a
Yes.
mixture of exponential large number of Gaussians. But can we say something about the second moment? I'm not sure that.
Because it's 0 means that the second moment will have like basically like we just drop n and that will be the variance, right?
Yeah.
So I think it will converge because our like if we really sample at random from here, then its second moment should be okay as well.
Yeah. Yeah. Agree.
Mm.
No, that's already not bad. So we have unbiased estimate with respect to the first and second moments.
Yes.
Do you understand correctly that if you know the hidden variables, numbers of your components, IDs of your components, then you can evaluate the density exactly? Like
No,
if you know from which Gaussian your variable
that's the thing
then you can evaluate the density exactly.
Yes, if we know, but
so you can sample. You can in MCMC. There is just like a trick when we can expand our space in variables, like add these latent variables and sample them also. Then you can evaluate your density exactly, and then you can just throw away these variables, and you will obtain like an estimate of your marginal distribution, like if you integrate it over these variables.
But you can obtain only the samples from the distribution, not
yes,
the
but you
density.
want only samples because we just need only samples from these distributions.
I'm not sure because
of beta, P, sigma. Yes.
But we're going to optimize likelihood with respect to pi and sigmas, and to do that we need to have access to the density function, not just to the samples of the distribution.
Okay.
But we are very interested if there is another way to fit this model, right? With respect to the hidden parameters, we would be very interested if we can. Yeah. So another trick would be the convolution from the Fourier theory, right? Because in the spectrum everything is pretty simple. We can just multiply, right? Because each of the things that we put in the linear combination, we know the analytical spectrum, right? So now just what the three formulas on the top, they have nothing to do with our specific prior. It's just the general definition of the characteristic function, right? And then we are actually working with an even function, so we can just write it as a half finite integral and a cosine, right? So that would be our way of calculating the PDF of z at a given point, right? And then we have because our I probably should rename to the same exactly the same linear combinations, but the characteristic function of a mixture is like a linear combination of the components of the mixture in this case. So the delta function is 1 and the normal distribution is this one. So for everything we put in the linear combination, it's also pretty trivial characteristic function. Then we can just multiply them, right? But
and this will correspond to what? To convolution?
To convolution.
And the convolution will correspond to the
yes.
summation?
Yes. Yes. So that's another kind of, it's a Fourier integral at a given point, right? Because we don't actually interested in the full distribution over, we are not interested in PDF, so this PDF across the entire like set of z values. We are interested in a given point to compute likelihood.
Yeah.
Right, so we don't want the like discrete Fourier transform. We just do we actually implemented this approach, and it was really, really nice. Sometimes normally outperforming the previous sampling approach with this one. But of course, it's pretty heavy because it involves the numeric differentiation, right? So at the end, we need to do this differentiation. We use the cubature, like numerical algorithm to integrate, and then to compute each data point, we actually do a product of, of course, the matrix was sparse, so each time it's around 2000 on average, but it's crazy amount of computations.
But wait a minute. After you multiply such characteristic functions, you'll again end up with an exponentially large number of items.
Not really, because here it's a number. This time is a number, and we actually do numeric integrations straight away. It's like we compute so this is no longer
at a particular two values.
Yes.
To your curvature.
Yes. So in this sense, it was a viable approach.
Nice.
Of course, insanely slow, but still faster than the previous one. And then there is a third idea, which was really difficult to get to, so it's probably a bit more involved. The previous two were kind of straightforward ways of attacking this problem. This one was I spent some time working on it. So we can actually make a moment like the kurtosis preserving approximation. We can preserve the fourth moment of the distribution. So we know the second and fourth, of course, the first and third moment are zero, because symmetric, and it's also not difficult to prove that third moment is also zero.
Third moment
yes. And
but here you have a mixture of Gaussians.
Of two. Just let's at this one let's just. So I think it's zero because the entire thing is symmetric. Just symmetry saves me in terms of convincing people where there is no skewness to the left or to the right in the distribution. It's also it's even. It's the sum of even random variables. I think should be even still.
Yeah.
So the two non-zero moments here is the second and the fourth, right? So we can do that for the betas, right? So beta here was the one with the prior mixture prior, the delta plus Gaussian, right? We can do that, and 3 is because that's a kurtosis of the Gaussian distribution, right? So there is no magic in terms of if it's a mixture, then the expectancy is a mixture as well, just by straightforward definition. So basically here it's 3 sigma beta fourth power because that's sorry, it should be 4. This one should be 4. It's a typo.
Yeah.
So then interestingly enough, in this case we can compute so if it's almost z that we looked before. I've dropped the plus epsilon. Let's just look at this one. So it's possible to derive these formulas for the expectancy, second and fourth moment, raw moment of the linear combinations of betas. I think we've only used the independency and zero mean variables in this case. So this probably, I didn't put, I think we won't have time to just go through all of the details, how do we prove this theorem, but it turns out that it's almost an excess kurtosis, right? Except excess kurtosis by definition is fourth row moment minus 3, right? I think that just minus 3, not minus 3 second row moment. So if it takes the fourth row moment minus 3 times the square of the second row moment, it feels like this is an excess kurtosis. Maybe people should have called this an excess kurtosis, but it's not, right? But let's just for the moment call it some kind of another excess kurtosis, right? It turns out that this thing is linear with respect to even in the case of the mixture, this e of the delta fourth power minus 3 times the square of the variance is cumulative and linear because the same thing goes here under summation. We can compute it exactly, the fourth moment of the complex distribution of the z score. Even though there is an exponential number of the things in the mixture, we still get the fourth moment exactly.
Yeah.
So must be some kind of, we can't be the first ones to write this fact, I think. But it was pretty interesting when we figure out that we actually don't, not only, the fact that we know the second moment is pretty straightforward, but the fact that we actually know the kurtosis, how heavy are the tails, and the heaviness of the tail is the main reason for us to bring here the mixture, actually. Mm-hmm. So we basically, that's another question I have on a discussion list later, is what other prior we can put, the prior with heavy tails, but kind of a bit less cumbersome to work with when we go for linear combinations. Heavy tail prior closed form under linear combinations. But let's put it for later, right? Once we know the second and the fourth moment, we can actually just try to come up with some parametric family of the distribution and try to match the parameters, so we end up with the same variance and kurtosis for that replacement distribution. It turns out that if you have a mixture of two normal, kind of the same analytical formulas, it's possible to find another weight in the mixture and another variance for the two-component Gaussian distribution in a way that will exactly preserve the second and the fourth moment for our distribution.
But is it one-dimensional?
It is one-dimensional, yeah. As was our previous z-score, right? It was just one number, one-dimensional
okay.
random variable.
But before we had a one-dimensional distribution which consisted of exponential
yes.
mixture
And
distribution.
in this second lemma, I mentioned that I can, for that complex distribution with an exponential number of mixtures, I can still find its second and fourth moment.
Right.
And now I would like to have another two-component distribution with exactly the same second and fourth moment.
You're trying to approximate the exponentially many number of components of mixture of Gaussians with a two-component mixture of Gaussians, where one component is fixed to be derive delta
yes,
zero,
yes.
and we have 3 degrees of freedom. Right?
Just 2 degrees of freedom. P1 because
okay.
P0 is 1 minus 2 and 2 parameters, two constraints.
And we are by definition have a zero mean.
Yes.
So and we have 2 moments. Yep.
Yes.
Second moment and fourth moment.
Yes.
2 degrees of freedom.
Yes. And it worked, right? So it turned out that there is no kind of corner cases as long as for any two numbers we can. And these numbers, I mean, these two numbers. These are the second and fourth moment. If we define the parameters pi1 and s in this specific way, then we end up with the known second and known fourth moment. So it's and you know, comparing to what we've had from before, all of the like either 20,000, a mixture of 20,000 Gaussians, right, with sampling approach, or this crazy numeric integration that involves computing this really large number of products and how numerically unstable is this product, and then a numeric integration which also has some kind of, and it's a numeric integration of the oscillating functions, so might converge, might not, and I think a theory on its own, right? This one was like, it's pretty nice, and it worked quite well, except still not good enough. So we use this approximation to find an initial, like once we fit based on log likelihood. We use this to come up with initial point in space, and then we go for the full solution with convolution. So this most precise solution, in order to like refine our parameters. It's nice to have different solutions to the, like, maybe we can like move to the log likelihood, the optimization part, right? So now we have a log likelihood. Now we have a way, like actually 3 ways to compute in log likelihood of our problem, right? And we can minimize. Basically, now we can our like work. Optimization is, we are not expert on optimization. What we end up with is the differential evolution algorithm, which we, like, I don't know, have you heard the differential evolution from before? It seems to be, I think the first choice for any kind of direct optimization would be the Nelder-Mead, right? The F-mean search in MATLAB, or the simplex method, right? Not the simplex method from the mathematical linear programming thing, but the simplex method in
zeroth order.
Yes, zero order Nelder-Mead, I think. Sorry for not saying it right. So, but that one required you to specify the initial point in space to start the optimization from, which is tricky, right? So there is another zero order method called differential evolution, which is really nice. You give it a region. It's more like it's a bit more of a global search method where you can, there is a set of, so in F-mean search, Nelder-Mead, it's n+1, that's your population size of the points in space that you use to optimize. Here it's more like a larger population in a global optimization algorithm. And so now we really numerically optimize our log likelihood function with respect to parameters. And we see that there is a pretty clear minimum in terms, for example, of pi1, right? So based on this simple log likelihood, we can minimize find some reasonable parameters. But it's noisy, right? So that's why I discussed the 3 solutions to this problem, because it's it's very hard to get. We have to, in our domain, we have to prove that something works in simulations. We generate data according to the assumptions of our model, and we have to prove that once we run the optimization on this data, we actually recover the unbiased estimates of the parameters. And this is the profile of the energy landscape of the log likelihood in 2D, right? Once we have pi and sigma beta and 1D, for example, as a log likelihood as a function of one parameter. And we can easily be stuck in the local minima, for example, this when it's noisy. So this data was from the sampling approach, right? So once you sample, obviously you introduce some noise to the energy landscape as well. Yes. Oh, thank you. Yes. Yes.
You optimize the log likelihood which you estimated somehow.
Yes.
And you estimated the very big mixture of Gaussians as a mixture of 2 Gaussians.
Yes.
So here you compute the value of log likelihood of these 2 Gaussians. Yes? Or these features?
Yes. Yeah.
Why are you sure that it's okay? Because it's not the function which you actually want to optimize.
Well, so let's go.
And I think it's very not your function, because if you have a very, very much number of Gaussians in your mixture, it's not the mixture of 2 of them.
Mm-hmm.
So why are you
Okay.
sure
So
that it's a good estimation?
So let's go back to the maybe I didn't explain overall what's going on. So we have 3 parameters in this model, just 3, right? And we have some observed data, but this data is not the actual parameters, right? There is the data is z-score, and I think whenever we have so we are isn't this? I thought it should be the straightforward approach. Once we have parameters and we have observing some data, and then we maximize the likelihood of observing the data given parameters.
The only thing that is
What's so?
here is W.
The W. Okay, sorry for that. The W is weight, and the weight is a number. It's a fixed. I see. So the W confused you because it reminds of the EM algorithm. The W we do not optimize. The W is a fixed, hard-coded. It's just. I put it here because it's a very good question. Why we need a weighted log likelihood?
Yeah.
But the weights are hard-coded.
Okay. So.
But the question was not about weights.
Yes.
Your likelihood is a mixture of exponential number of Gaussians, yes?
So the yeah. Yes.
And you estimate it with a mixture of 2 Gaussians.
Yes.
Are you sure that this estimation is good?
Okay. Now I understand. That's good.
Of the different functions.
Of course. So it's not perfect. Definitely, same as as any approximation. The only thing I know about this approximation is that it preserves the moments, right? It preserves 2 moments. Some people in this domain just preserve the second moment, the expectancy. We preserve 2 of them, but we don't know. It's about we run in extensive simulations. It's 20 pages in the supplement, and we've across many parameters, this approximation was reasonable, but only reasonable to serve as a supplementary helper to find initial point for. So, what we know is this is an exact solution, right? So we've used for final optimization, we've used the convolution. So the Gaussian was a nice trick to speed up. It's nice to be able to compute, it's approximately 160 times faster. It's cost around 10 minutes to compute this thing once, on a large server, and this one takes seconds. So that was the reason for a performance improvement and some kind of ability to have a flexible fit procedure in this case.
Could you please comment why don't you have here a gradient optimization?
Yes. So I think this slide would be a good example. you see, it was noisy, right, in some case, and like finding the gradients of the noisy function would be like even more noisy. So we've tried that, but just we were not able to get any improvement in like save time by using like first order or second order method, because our estimates of the gradient were noisy. Would that be a...
So you have an estimation of a function that you want to optimize.
Mhm.
That is noisy.
So our log likelihood is noisy, yes, right? Because like look here, so this approach requires some kind of arbitrary number of samples. Depending on how many samples we have, we can make it quite smooth, right? And for example, this was pretty smooth surface, right? So this one was specifically computed with the first approach. It is pretty smooth, but if I can try to zoom in, it's a very, like you see, it's first there are some linear combination of parameters that seems to be not well identified, right? This kind of black region throughout, and then the noise was quite important. So it was difficult for the optimizer method. So anyway, the first one, the reason for the noise is, in this case, it's a finite number of samples in our approximation. Here is a numeric integration. Depending on parameter, the numeric integration here with kubature might take a slightly different route, right? And it's noisy. This one is absolutely smooth. However, it's an approximation, which is not good enough. So that's why we are optimizing the noisy log likelihood function. But, you know, so that's like I know it's a hard problem. We really spent just two years trying to figure out at least some solutions that work, right? It's an important problem because like that's the only genetic data we have, and you see that like we start with 2 terabytes on disk of the data. The genetic matrix is really large scale genotype data, 2 terabytes, and now we are trying to estimate 3 parameters out of this data. And despite like different 3 approaches to computing the likelihood function, it still doesn't really so shiny. I would be happy to give an overview of the properly Bayesian methods that do the same, right? I mean, they also struggle with similar problems with very slow convergence, long time to compute.
I think we'll at least try to suggest you one presumably quite efficient, computationally efficient technique for solving the problem.
Yeah.
I also have a question about motivation. Maybe I didn't get in the beginning. What we will do with that estimated P and C?
Good question. So first, it's very interesting from the domain perspective to interpret these numbers. The pi is a polygenicity parameter. It's like, we know that some traits are Mendelian, like it's one gene explaining everything. Some traits are crazy polygenic, like schizophrenia. But pi is a very interesting quantitative measure that we can speak about. The larger pi, the more genes is involved, and that's interesting for people to compare. And the sigma beta is the effect size. It's interesting, like, is this big or small effects involved? And the product of these two is the heritability. So we can, from this model, read out, is this trait heritable, like by 80% or by 1%? If it's 1, probably it's not interesting to study, right? And the last one, the sigma 0, is also interesting. But and another thing, like let's look at, there are some posterior integrals. Once we've fitted the model and we know that the model kind of describes the data, we can estimate, like sort of, that's why this slide may be the reason why I think it might be interesting to discuss it in this group, because like we do use a very basic Bayesian posterior function, because okay, now what we have the distribution over z, right? We have an observation of z, and we are interested in some posterior, let's say, expectancy of delta square, right? So like we're interested in this quantity, which is basically the proportional to the expectancy of delta to some power, like the 1 or 2, given our observation, right? So for that, we need a model, right? In order to make it happen, we need the probability distribution over z and delta, kind of like as a joint distribution, right? And for example, this parameter was extremely interesting in a field.
Yeah, expectancy of delta to the power of q.
Yeah.
You mean expectation?
It could be, yes.
With respect to what distribution? To the posterior distribution over delta? Or not?
Yes.
You
Yes.
wrote here up to constant.
Up to constant.
Multiple with constant,
Yeah.
it's true.
Yes. Sorry. Sorry for that. I'll try to speak in the microphone. And just I want to share this trick. You know, I've integrated last time at the university. It's not that much effort I spend in integration. And this, to me, looks like pretty difficult. Of course, we won't go through derivation. And I can tell you, actually, I've never done that myself. I never derived these formulas. I just feed them into the Mathematica, the Wolfram Mathematica. It's so good at doing these integrals, right? It's really just amazing.
What is the quantity of S?
So
why do we need it?
So yeah, I don't want to overwhelm you. Like the S quantity, this ratio is telling us. So you see, it's about large z-scores, right? For large z-scores, what's the fraction of z that was large, right? And it's basically the S number is how much we've discovered with our genetic studies.
Mm-hmm.
So this conceptually is if we just drop the dependency on the sample size, but obviously we have it, right? Because z is a function of n, which is the number of participants in the study. So S as a function of n tells us at a sample size of 10 times larger, 100 times larger,
Okay.
once we get 10 million, 100 million people, how much would we be able to explain, right? And like this is, we feed the model and now we estimate the future of the field in a few years from now, right? So that was, and you see it's very interesting that once we, you remember Z was really complex distribution. It's the one with the exponentially large number of components. But because one of our approaches was that's simple to do it as a mixture, that's sum as a mixture, right? A mixture of normals, right? Then we can actually estimate this both denominator and numerator under this approximation, right? So it feels like kind of going down to this shape of the approximation on Z distribution is really fruitful for us, because we can under this model we can estimate a lot of posterior functions, right? So and that's just one of them. I had a bit more complex things for 2D, for really once, that was all 1D, right? I think I will speed up a little bit because we need some time for the discussion at the end. So the paper here was actually about 2D. I started with 1D because you've seen it was kind of complex enough discussion, but we are interested in the cross-trait analysis one we have for each marker Z1 in one trait, let's say schizophrenia, and Z2 in the bipolar disorder, and we are interested in how they overlap. So I just skip through some figures that just to give you the feel of the data. In simulations, if the betas look like that, right, then Z will look like in the bottom here. So it's now it's two traits, right? So probably hard to switch from just having one per variant to two. But the idea was, let's say if we have the genetic effects completely independent, like if something has an effect on one trait, it doesn't have an effect on the other trait, and the opposite, right? If something has, right? Then what we will observe in the space, this is hidden, right? We don't know real genetics, right? This is the top line here is our simulations data. But then under these assumptions, that's what we will observe as a Z data that we can optimize for, right? And you see it's pretty noisy. In reality, it looks like that. It's really I also like the visual. The visual appeal is like a star or something. But basically, the Z-scores for schizophrenia right now are between -10 and 10, right? And the bulk of them are really uniformly distributed normal, noise, just noise. And if we plot them, this is a density heat map between what we observe in the 1.3 million education study on education attainment versus 100,000 study in schizophrenia. Yes, it seems that it's a white noise, right? Kind of not much data. Some outliers, right? So it's what's in the corner, that's pretty interesting, but that's a little bit difficult to. I don't know. Yeah, are you familiar with the QQ plot as a concept? In statistics, that's pretty quantile plots to assess whether two distributions have similar shape.
It's better to remind.
Yeah. So.
Not everybody's.
Yeah. So this is our synthetic data, and it's not directly the very standard QQ plot will look something like that. You have Z1, you have. Probably not the best. In statistics, for example, you typically need to test. Okay, let me do that. I think something the opposite, like this. So yes, yeah. How do Yeah. So, for example, in statistics, once you do the standard linear model, it's important to test that residuals of a linear model are normally distributed, right? And you can probably do a Kolmogorov-Smirnov test for normality, but on a large sample, it will always tell you, oh, yes, it's a little bit not normal, right? So what people do is they make a QQ plot. So they see the difference between the observed distribution of the residuals versus the expected under the true assumptions, right, which is normal. So, and then the plots look approximately like that. It's a quantile plot. So one of them, I don't know what people normally write on the axis. One of them is the actual z that they observe, and another one is an expected quantile under null. So they sort the values, and yeah, probably not the best explanation. Anyway, so the standard one is on the natural axis, just z-score versus z, right? This one is in log axis, so probably it'll be a little bit hard to understand what we plot the logarithm of the p-value that we observe versus the logarithm of the expected p-value under null, right? So if there is no signal, it will be flat, like kind of following the null. And the fact that it deviates, it means that in our sample we have an excess of heavy z-scores, right? Because if it goes up, like in all of the plots here, and there are really very bad colors here, but the blue one is the real data, and the red one is expected from the model, because we can also plot according to our model of the PDF of z, how would it look like, right? So, and effectively, once we feed the parameters with log likelihood, we can kind of see that the curve actually starts describing the actual data, right? So the thing here is about, we played with a pi parameter, and we play with the h or sigma beta parameter. You see, the larger the sigma beta, the further it is from the null. Like low sigma beta, it follows the null. The larger it is, the more signal there is in the data.
And
right?
what's the difference between the columns?
Between the columns is different pi. So this is pi of 1%, pi of 0.1%, and 10 to the minus 5, pi 1. Right? And we actually, so this
pi is the probability of non-degenerate.
Yes, and like that's this is from synthetic data, but we've seen like all these cases in the real data. So, for example, the pi is is very nice shape from here. Pi tells us where it will start to bend, right? Basically, right? Except there is this linear combination, right? So we put together through the linear combinations, the actual proportion of things associated, because it's in Z space, right? I can go back into the concept.
What was the dimensionality?
Number of SNPs?
No, dimensionality of Z.
Yes. In this case, 11 million. So we have 11 million statistical tests, basically. And a lot of them are null. And in the tail, they're not null. So, yeah, that was like a nice way to look at this data in terms of like, it's not. It looks pretty noisy, but in the end there is a small tail that is definitely a non-noise.
What is the dashed line here?
The dashed line is null. That would be under null. It's what we just plot as a reference. So the dashed line in this one, right? The dashed line is like, you know, that if we have a collection of the p-values under H null. At H0, they will be uniformly distributed on 0, 1, right? And if you look at the z-scores, if you have a collection of z-scores under H null, they will be distributed as a normal, right? Yeah. So that is the dashed line is how would the QQ plot looks like under null? Under H null for each of the tests.
Under normal distribution?
Under normal distribution, yes. Yeah. So yes, at this point we can move to the kind of discussion part of this, kind of what we would like to do next with this thing. The first paper is out. Now I'm interested in like, is there a better prior? Because you've seen the 3 solutions we put to the prior. They are noisy, they are costly, they are not really good, even though we spend some effort on it. So what we are looking after basically is a heavy tails, right? It's a heavy tail probabilistic function that is closed under linear combinations. Like I'm not probably forgotten everything I was told but I know the normals are closed, but they are not heavy tails. But so yeah, if you can think of any prior that would replace our strange mixture of delta and the Gaussians, that would be interesting. Another one is we're actually very interested to model the dependencies between different betas, right? So for us, they were independent. The different betas were like independent in terms of like as random variables. But like first, we already work at like a lot more complex prior when it's a mixture of 4 components and it's different sigmas and could be different pi's. And the sigmas could be modulated, but whatever we know about the human genome, right? And the plots I've shown you from the 2D, we actually modeled as a mixture of 4 Gaussians, some kind of a little bit degenerative, and then the full Gaussian component. So that's one of the, we are playing with very different priors here. And one specific topic is, can we model the dependency between betas? Because like we know that the SNPs, basically the SNPs or these genetic variants belonging to the same, let's say, genetic pathway will behave similarly, right? There is so it, like bioinformatics is such a large field that they know a lot about the connections between different parts of the genome. For example, there is a pathway of neuronal differentiation. These are all genes involved in like whatever is going on with neurons when they split and kind of mature. Right, so we would like to put that as our prior on beta, and that's why. So maybe correct me if I'm wrong by saying that we had a prior on beta, right? So it's not a prior on meta parameters, but like we have our domain knowledge about how beta looks like. One of the things was that it's a heavy tail distribution, right? So that's why our prior knowledge was translated into the delta function plus Gaussian. But we would like to put more things, and like what I'm thinking here is like more like, I don't know, for example, in the topic modeling, right? When we model the relationship between documents and these collections, it's nice to model the like authors, right? If someone cites someone else, right, then there is a relationship between these two entities. So here we also would like to model the dependencies between betas. Because our prior so far, it's not the interdependency between beta. It's like we can say that, okay, in certain region, betas should have larger effect, right? That we can model with. So that's my project right now. I have the enrichment. In given regions, we expect betas to have larger pi or larger sigma, right? But what if we can say that these two betas are either both small or both large? That is interesting. And also you've seen that basically the posterior that I mentioned was about the deltas, right? So if you say that z is equal to some kind of delta plus epsilon, right? Sorry, I don't have another slide, but so we found the expectancy of the delta, expectancy of the genetic component. But we didn't work out this correlation structure, the A, right, across SNPs. So that would be remember, what we actually kind of interested in here is to compute in the betas themselves, right? With what I've shown today, we didn't address this question in any way. We said we put a prior on betas, and then we can estimate the parameters of the prior, because it's like either helps us to answer some questions related to the domain, or we can basically interpret these parameters, the pi, the sigma beta, the sigma 0, but we did not in any way address the question, what are the actual betas themselves, right? So we would like to do this kind of estimate A pseudo-inverse times Y, or times, here the G pseudo-inverse times Y, right? At this scale, can we do that actually? Like some people say they can, but their tools are not quite promising, and that's like if I have a chance to talk with an experts in the linear, like huge linear algebra, how do they approach this problem? It would be interesting, because it's a block matrix, right? So maybe we can do something because see that's a valid problem, right? We can estimate, given our observation of z, we can probably like formulate this problem to estimate the beta itself, but solving it involves the pseudo-inverse, and that one is really hard to do. So another big topic in this domain was mixed effect models, because our model today was just a fixed random effects, right? We had only the random effects beta without zero, without a fixed mean, right? All our Gaussians throughout the whole presentation are zero mean, right? But it's definitely interesting to introduce some fixed effect in the model. Let's say, okay, what if we have a genetic variant that is, we would like to like model, is it increasing the height? Is it decreasing the height specifically for that given variant? Like, can we do something then? Because this whole domain is called mixed effect models, right? They have the restricted likelihood, restricted max likelihood, right? Because the problem is, so who just heard of the restricted max likelihood as a okay, then probably it's a separate, completely separate thing. You know, there's so many things going on in statistics, but it seems to be a huge domain with like 40 years back they had an algorithm called average information, and since then people come out with a whole bunch of other algorithms for fitting this. Yeah, so optimization is another topic. Right? So basically I'm just trying to brainstorm, right, since I came and bother you with this really kind of plenty of mathematical questions and details, and like what are the potential feedback points we can discuss. Other than that, actually, I can maybe mention how I can be helpful, because I think I'm pretty good at this last part is computing. So just to implement this stuff, we used a mix of Python and C++, and in C++ we definitely had to go for the like open API to do it concurrently across threads, and then we had just numeric integration, just what we plugged in, but we spent a fair bit of time compressing and storing this matrix. Originally it was like 70 G in memory. Doesn't really work on our clusters with 64, so we compressed it down to 10 using small integer optimizations and etc. And like just working with some high performance instruction sets, etc. is pretty interesting. So if I really like these computational things. So I first don't want to go into detail right now, but it was nice. We started with MATLAB. Everything was coded in MATLAB originally, and then it just doesn't work. It is so custom and specific. Like MATLAB is a matrix laboratory. If you stay with matrix, it's really good. It has a lot of good algorithms. But if you have to code down to hardware, that's not that fun. And of course, we didn't touch on the beta prediction, because if we have the beta hat, even the one that is naive, right? The beta hat that is just a correlation of the naive Bayes classifier is what 60% of the publication in this field are from the naive Bayes classifier, which people know under the name of the polygenic risk score. It's the estimates for a given variant that we completely forget everything except for a given genetic variant, look for the correlation, come up with a beta hat, that how this particular variant correlates with a phenotype. And then we disregard the correlation structure, everything just in a very naive classifier way, sum them together, right? Still have some predictive performance, and that's what people use for all of that. That's kind of the mainstream genetic studies. And with this beta priors and beta, maybe we can do the better estimation and better prediction, which is helpful. So that was the models, and with that, I just acknowledge my colleagues. We are a pretty large team, and this is NORMENT part, of course. Not everyone is working on genetics. You see, this is a large family. Right? For actually, some of them are really real family, because my wife is also here. But yeah, we also have colleagues on the other side of Atlantic, and the actual the really guru who inspired this particular mixer thing was Anders Dale in the University of California in San Diego. We worked really hard with him, and just together tackling these nasty mathematical problems for us. So yeah, I think that's it. Do you have some questions either for this discussion that I put or back to the slides? Like, yeah, any questions? Yes.
You mentioned that you want to optimize your code in a way. Which compiler are you using now?
I've ended up with just GCC.
Well,
Yeah.
GCC use Intel compiler can do 2 times.
Yes. I've been on the trainings from Intel, and they don't say that anymore.
What?
I've been on trainings with Intel, and they don't say that anymore. So for some things, again, yes, it could be faster, but
I actually tested it myself. It's 2 times faster.
I think it depends on. I also tested on some. It depends on the code, right?
It depends on
For
the code and the optimization you're using, and if you want to really push it to the max, use until intrinsics. You
Mhm.
can
Mhm.
utilize your CPU, and you can use ATX 512.
Not available in our cluster. Sorry. Yeah. Yeah.
But anyhow, using Intel compiler.
But your code also, right, should support that, right? So it's again, it's always a balance. Like, I have a limited time I can spend on it, right? So I know I can probably tweak maybe 20% more of performance. But yeah, you're right. That's a good point. I had on the list to. I need to see if Intel straightaway is available on our cluster. Yeah. I know, but that's installing it, and yeah, we again. I think I've also, like, for this stuff I've tried, for example, using MKL for the things I could. I've tried to use MKL. It wasn't at my faster. Yes, yeah.
Well, yeah, but you need Intel compiler in order to optimize to the.
No, but if you call MKL, right? It's if it's dynamically linked, then what's again? But it's a good point. We could try and see. It's after all, it's an empirical question, right? It's yeah. I'm aware of the Intel compiler. It's definitely a good thing, but.
Yeah. And another thing, if you want to do it like code easily like Python with the same performance you will get from C or C++, use Julia.
Yes, so my colleague was, I think, 1 year he coded everything in Julia. I think at the end he switched to C because it was, some libraries are not available. The good thing is Python, it's so, like the NumPy, CPy, everything is already available. Julia is an excellent thing, but so we don't mind this mix. We don't mind having the core performance modules in C++, and then it's like integrated into Python. So Python is for high level, like data logistics and like smart things like fitting the model, and then the actual hardcore stuff is in C++. Yeah, but it's interesting. I like code. Everything is public, so this stuff is public. It's all the research code, the research code and the production code. But yeah.
Well, I have a question about, does anybody try to learn a nonlinear model? Because, well, currently in data analysis, almost nobody learns linear model. They were somehow compromised. They are not able, they're too weak to capture the real dependency in our data. And here you learn linear model.
Yes, that's a good, absolutely. I've been thinking why. Why, like, you look at the papers and they don't. So in domains it's called epistasis, the interaction term between the different genomic locations. So, and one of them is like this structure. You would definitely expect nonlinear effects within the block, within one block, but probably not across blocks, because if there is a genetic effect that is nonlinear, there's like different chromosomes, right? Things interacting on different chromosomes is difficult because they independently inherited. So what I have, I get from my mother and from my father. If there was something important biologically, right, I should have passed it to the next generation. But the fact that the chromosome in our body is this continuous 1D structure that randomly goes to the next generation, it must break whatever there is, whatever dependency across between features we have. If it breaks across generations, then probably it would not survive the evolution, right? So there is, explaining that is, I think in order to have an interaction, you have some kind of unit that survives when it goes from the parents to kids, right? Otherwise is, so we definitely are interested in interactions within one block, because the fact that within things that are like highly correlated and go together, like from from parents to kids, that is interesting. On a different chromosome, so remotely on one chromosome, I feel linear model could be the right biological.
But anyway, does there exist any paper that actually try to learn this nonlinearity?
Not to my I can look it up, the exact papers, because the term is in the field. People use it in not in this complex. So this whole thing was about complex polygenic disorders when the individual effects are small, but they are scattered across many genes. If you talk about the Mendelian traits, when like there is a very known functional broken protein that causes very like severe dysfunctioning, then I think people do study the nonlinear, but not throughout the whole genome, like in this case. For some, like partially the level, like this model is hard enough to learn, so we kind of don't jump on a more complex problem before we feel like this one is under control. Another thing is this biological reasoning that it's only the things that go together from parents to kids that maybe you can, in other words, let's say there is like some interactions that gives, if you speak of the height, right, how tall is someone, right, if it's not a linear combinations of the gene I have, right, but then it's actually, the genes from me to my daughter goes independently, right, so how it kind of don't match the non-linear. It's only, so whatever is linear, that preserves, right? If there is something giving me an extra centimeter of height, right? And some other parts of the chromosome that gives another 2, right? And then they go independent. Whether they go or not, they still kind of additively add to the height of the next, of the offspring. So I think there is a pretty solid biological justification for the linear model, except when we talk for the one block. Or if you talk, yeah. But
okay. Are there any more questions?
Speaking about a more heavy-tailed prior that is, as you said, closed under linear transformation, right? Well, I think that Cauchy is closed under linear transformation.
Cauchy, Cauchy distribution.
It's like the most heavy-tailed distribution.
Is it parametric?
Yes, of course. And it has a much more simple parametric form than even normal distribution.
You can try. Is it 1 or 2 parameters? Just
well, shift and
yes, shift and like
scale.
Yeah. So for us, the shift would be 0, right?
Yeah.
Shift would be 0, so it's 1, right? And that's so. I think here it was quite important that it's two parameters. So one affects like how heavy, how large is kurtosis versus how much variance do we have, because these are 2 independent properties that.
Kurtosis and variance.
Yeah. So in some sense, this was.
What about using the mixture of delta function plus Cauchy?
We could try it, because now the mass that represented was pretty flexible to that. So we can try. We can try. Yes.
The only problem is that for Cauchy we do not. That doesn't exist any in the first moment. Not talking about the second one. So on. No moments exist.
Yes. Yeah.
They also took
Okay. So that I.
Okay, well, I think that that's at least worth discussing.
And just if you're like, I'm happy to share slides, and if you would like to, everything is written down as a supplement of this paper. It's actually, some things are cumbersome because it's the first time in 3 years I was given a chance to speak about the actual math behind it, because it's always like presenting it to like the domain expert. It's not about math, it's about the interpretation and everything, right? So therefore everything is in supplement. There is nothing of what we discussed into the main text. Like this is a paper, the supplement has all of the stuff, if you're interested in. Thank you.
Okay, so let us thank the speaker for a very interesting talk.